Hello and welcome to this talk about how calcium carbonates form and why we should care. My name is Juan Diego Rodriguez-Blanco and I am a lecturer at Trinity College Dublin, Ireland. I want to start this talk by showing you this picture of Mons Clint. This is a place which is very beautiful because these are white cliffs made of chalk, made of calcite, made of calcium carbonate. And also this picture showing you a biotic calcium carbonate. This is typical calcium carbonate that you can form during industrial crystallization. These are crystals which are completely different to biominerals, but these are like the end members of this big equation because carbonate crystallization is fascinating. Carbonates are minerals that can have a very wide chemical variability. They can form by many different processes and they can be found at many different scales. And I want to show you here in the big puzzle of geochemistry how calcium carbonates can be present at scales that range from the atomic to the light year. So I'm going to start with the small ones, atomic and nanoscale. So at the nanoscale, you can find calcium carbonate usually as amorphous calcium carbonates. These are precursors of crystalline phases, and these carbonates are metastable. Sometimes they can have ordered domains that are have the sizes of a few angstroms, but they transform into crystalline carbonates, which are the carbonates that you know, the minerals that you know, like calcite, batrite, aragonite, dolomite. Okay, these carbonates are like the main characters. They are the building blocks of biomineralisation. Okay, and at the same time, they are the building blocks of rocks that are widely distributed on Earth, from chalk, limestone, marble, and many more. Okay, and these rocks are essential for us. They are essential because they are useful materials. They have impact to everyday life. They are essential for many industrial processes, from construction to paints, food production, health, paper, and many more. And also, at the same time, they are key in science because calcium carbonate or calcium magnesium carbonate rocks can help us to understand past environmental conditions. So we can know the ambient formation of these rocks by studying the trace chemistry in these minerals. Okay? This links to many processes which were happening in the past and happens now in the present and will happen in the future of Earth. And of course, they have a very important role in the carbon cycle. It's impossible to understand climate change, CO2 sequestration without taking into account all carbonate crystallization. Also, carbonates are found far from Earth. They can be found in other planets, for example, planet Mars. There are rovers on Mars that are now studying carbonate deposits, and also in many other places far away which are more difficult to reach, in far deep space. For example, we know that there are carbonate granules in nebulas. They have been detected by using infrared spectroscopy. So they are important as building blocks of planets. One of the most important questions in carbonate mineralogy and geochemistry is to understand how carbonates form. Because, for example, in biomineralization processes, we know that this formation is taking place in very small, very confined environments under the presence of a lot of organics, and also that happens very quickly, sometimes in a matter of seconds or minutes, and it's very difficult to follow these reactions. Okay, and understanding these crystallization processes will have many implications and will help us to understand many processes happening at the global scale. For example, that will be enormously useful to understand the evolution of ocean chemistry, to understand the problem of ocean acidification or the fate of the pollutants that we dispose in the oceans nowadays, or the multiple ramifications of carbon cycle. But also, it will be fantastic for industry because it will be easier to produce calcium carbonates or carbonates with different shapes and targeted properties. Okay, and the problem is that we don't know very well how minerals and in particular how carbonates form and transform. We know that from supersaturated solutions we can go to final crystals, but sometimes that may happen via classical pathway by nucleating crystals and growing them, and sometimes that can happen via the formation of prenucleation clusters, amorphous precursors, which later crystallize. This is by a non-classical pathway. And these processes are very quick. They can happen extremely rapidly, and they are very difficult to follow. Okay, here I'm showing you a diagram summarizing the multiple ways of producing pure calcite. Okay, this is based on laboratory research. And here what you can see is a supersaturated calcium carbonate solution, and we go from lower to higher supersaturation, and at the end we produce calcite. This calcite has different sizes, shapes, and surface properties, but calcite can form via many different mechanisms and pathways. For example, they can be produced directly from the calcium carbonate solution or via an amorphous phase, and sometimes via different metastable calcium carbonate polymorphs. Some of these reactions can be extremely rapid. And here in this talk, what I'm going to focus is on this area, on the amorphous calcium carbonate. Amorphous calcium carbonate is not a compound that has been recently discovered. It has been known since many decades, more than 100 years. For example, it is known that it happens abiotically and also biotically, for example, during the crystallization of granules of calcium carbonate produced by earthworms. This is something that Darwin discovered. Okay? And in particular, amorphous calcium carbonate happens in many living organisms, from sea urchins to plant leaves, earthworms. Also, it happens abiotically in deposits in speleothems, and it's being produced by fish and also crustaceans, bacteria, and many other organisms. So it's not an uncommon phase. It happens that the amorphous calcium carbonate, which is produced in the lab, is metastable. It's highly metastable. It has a very short lifetime, only a few minutes, usually 1-2 minutes at ambient temperature. But the biogenic amorphous calcium carbonate has a longer lifetime because it dehydrates very slowly, and sometimes it can have a lifetime of months. That happens because this amorphous phase is produced in a biological environment, inside a membrane environment, where the activity of water is lower and there is much higher concentration of organics. So this amorphous phase requires higher supersaturation compared to crystalline calcium carbonates, and usually ions like phosphate or magnesium or organics can stabilize this phase. Otherwise, what happens is that the crystallization of this amorphous phase can be very fast and happens in a matter of seconds. And that's because this amorphous phase is considered to have a structure similar to this. Channels that contain carbonate and water, and also calcium ions. Okay, it's not a completely homogeneous structure, but it has this kind of channel structure. Okay, so I'm going to show you how we can follow calcium carbonate crystallization reactions, or in general carbonate crystallization reactions. So for this purpose, we combine classical experiments in the laboratory with synchrotron-based experiments. So what we do in the laboratory is, for example, to mix solutions, to take samples, and to characterize these samples with different solid states, spectroscopic and microscopic techniques. And then, once we understand more or less what is happening in our experiments, we do these same experiments in synchrotron facilities. These synchrotron facilities have a very good advantage, and it is like there we have very intense X-ray beams, and we can follow these experiments in situ and in real time. We don't need to take samples. And we use facilities like, for example, Diamond Light Source in the UK or Argonne National Labs in the USA, in Chicago. And the advantage of this is that with these very intense X-ray beams, where we select one station, and there what we do is to set up the experiment, as you can see in the image here below, and we obtain data every second, or even at times time resolution which is lower than a second. So this is a very fast data collection. We can see what is happening every second, even lower times. And the information we get is very useful because we get particle size and shape, the evolution of particle size and shape with time, and also the evolution of the crystallization. So we know which crystals are forming and transforming in our experiments. So from here we can get very useful mechanistic and kinetic data. So we can get the kinetics of these reactions and we can understand the mechanisms of calcium carbonate or carbonates in general crystallization. So typical techniques, for example, this is energy dispersive X-ray diffraction, which is similar to conventional X-ray diffraction, but the only difference is that we use white radiation. White X-ray radiation, and we have different detectors to detect what is happening in our experiments where you have crystals growing in solution. Also, sometimes we use hydrothermal crystallization. What we do is to mix the solutions at ambient temperature in a Teflon reactor, and we put this Teflon reactor inside an autoclave. This autoclave can withstand temperatures which are usually not higher than 220. The X-rays go through the reactor and we can follow this hydrothermal crystallization process. Later on, what we do is we take the sample and we can study the sample with, for example, scanning electron microscopy or other techniques. Okay, and usually what we can do is to obtain multiple data at the same time simultaneously. For example, we can obtain scattering and diffraction data simultaneously. Sometimes we use something named a stop flow system. It's a device that can mix solutions very quickly in less than a second, and we can follow the reactions extremely quickly by using the synchrotron light. So these techniques, SAX, which stands for small angle X-ray scattering, and WAX, which means wide angle X-ray scattering, give us particle size and shape, the first one, and structural data, X-ray diffraction, the second one. Here you have a picture showing how a typical experiment in the synchrotron looks like. What you can see here is the stop flow system that can mix these solutions very quickly. The X-ray beam follows this path and the beam goes through a capillary where our reaction is taking place. And we have here one detector, the wide angle X-ray scattering detector, and here the SAX detector, the small angle X-ray scattering. So as I told you, wax is like X-ray diffraction and this is scattering. Okay, and by using this method we can follow all these reactions which are very quick. Also we use another technique, it's named pair distribution function, and it's usually used for the analysis of powders, although we have done sometimes in situ and real-time experiments. But here, what we can do with this technique is to understand the nature of the amorphous phases. We can characterize the amorphous phases. And here what we have is the samples in capillaries. Sometimes these capillaries can contain some water, and the X-rays goes to these capillaries, and what you see here, this screen is in fact the detector. Okay. So now I'm going to focus on the main factors that affect calcium carbonate formation. I'm going to give you three main ideas. Okay, the first factor is the starting pH. So I'm going to give you one example with the pure system, with only calcium and carbonate. And if you want to make calcite, you can think that you can make two solutions and mix them. You can make a calcium chloride solution and a sodium carbonate solution, but you can mix them in two different ways. You could add the calcium chloride to the sodium carbonate solution, or you could add the sodium carbonate solution to the calcium chloride solution. I mean, you can add A to B or B to A. Okay, and there is a difference here, because if you are using mixing A, that means adding the calcium chloride to the sodium carbonate solution, what happens is that you start with a very basic pH, because the pH of a sodium carbonate solution is around 11. Okay, and if you're adding the sodium carbonate solution to the calcium chloride solution, you start with a neutral starting pH, because the pH of a calcium chloride solution is close to 7. And the consequences of this mixing are completely different. I'm going to show you what happens first when you're doing experiment 1, when you're adding the calcium chloride solution to the sodium carbonate solution. So what happens here is that you see here this diagram, this is time. Here you have 50 seconds, almost 2 minutes. So in 1 minute, here what is happening is that you can see amorphous calcium carbonate decaying and forming one mineral named baterite. Baterite is a calcium carbonate polymorph. Here, This axis represents 2 theta. If you don't know what is this, you only have to know one piece of information. Depending on the position of this peak, you can know which mineral you have. This is coming from X-ray diffraction, from the Bragg law, okay? And when you have no peaks, that means that you have an amorphous phase, or you could have only water. When you have peaks, that means that you have a crystalline solid, okay? So here what you see is that you are forming an amorphous phase at the beginning, and this amorphous phase is transformed very quickly in a matter of seconds into a crystalline phase. So what we see is that we go from the amorphous phase to this crystalline phase named batrite. Batrite consists of spheres that are made of nanoparticles. These nanoparticles are crystalline, okay? And that happens after the breakdown of this amorphous calcium carbonate in a matter of seconds, in less than 2 minutes. Later on with time, what happens is that this batrite slowly transforms into calcite. So what you see here is one peak of batrite, another peak of batrite, and these peaks are going down while the calcite peaks are increasing. So at the very beginning you have the amorphous phase, then batrite, and in a matter of hours this batrite decays, it slowly dissolves and transforms into calcite. So these are 2 very different stages that can take a while. I mean, at 7.5 degrees C, this almost takes 1 day. At ambient temperature, this could take 6 hours. So the crystallization of calcite will take this amount of time when you are adding a calcium chloride solution to a sodium carbonate solution. Okay, so this is a summary of the same thing. What you see here, this alpha means degree of reaction. This is like saying 0% on the bottom and 100% of solid. And what you have is quick formation of batrite, these black dots, and then slow decay of batrite and slow crystallization of calcite. 2 stages that in total can take a little bit less than a day. Okay, so we go from amorphous phase to batrite and then calcite. Remember, this is mixing A, in which we have the calcium chloride solution added to the sodium carbonate solution. So we are starting with a very basic pH. In a different way, this is the same. Degree of reaction, 100%, 0%. You see here the quick breakdown of the amorphous phase, the quick increase of batrite, the quick crystallization of batrite, that once has crystallized, slowly starts to dissolve and transforms into calcite very slowly. Check time here because this time is not linear, this is logarithmic. Okay, so these are the mechanisms of formation. So dehydration of amorphous calcium carbonate, growth of batrite via spherulitic growth, and then slow dissolution and recrystallization of batrite and crystallization of calcite. Okay, so this batrite formation happens via spherulitic growth. This is a mechanism of constant nucleation. So in this case, you're not growing a crystal. What you are producing are spheres or pseudospheres that are produced of multiple crystals via constant nucleation of nanoparticles on the surface of another nanoparticle. So you have an aggregate made of nanoparticles. This is how scanning electron microscopy shows this process. This is amorphous calcium carbonate, quick crystallization of batrite, and then the dissolution of batrite transforming this batrite into calcite. So ACC, batrite calcite, starting from a basic pH. So first stage, very quick, in a few minutes. Second stage, much more slower, and takes a few hours. Okay, so let's see now what happens when you add the sodium carbonate solution into the calcium chloride solution. In this case, we start with a neutral pH. Okay, so the process here is completely different because what happens is that you form an amorphous phase that breaks down and transforms directly into calcite. Okay, this is at 5 degrees C. We use low temperatures to slow down the kinetics of crystallization, and what we see here is that calcite directly forms from the amorphous calcium carbonate. So in this case, we go from the amorphous calcium carbonate directly to spherulitic calcite. Alright, and this only happens in a matter of a few minutes. It's a very quick process. So what we see here is what is shown in this red line. While in the first case we were forming amorphous calcium carbonate, then batrite, and then calcite, and this is what is shown in this picture at the bottom. Then in the second case we have this amorphous phase that breaks down and transforms directly into calcite. So the solid looks completely different, and it looks completely different because it crystallizes via spherulitic growth. Here we have calcite formed via dissolution and recrystallization, and here calcite formed via spherulitic growth. Therefore, the surface properties of these crystals are completely different, considering that they have the same composition and the same structure, and they were formed from the same fluids, but only this mixing difference and this difference starting pH can completely change the mechanism of formation. So why is this happening? Well, because of one simple thing. When you see how we mix aqueous solutions, we think that we mix them very quickly, but mixing is never perfect. What I mean is that something which is happening in a matter of milliseconds is something that looks very quick for us, but for crystallization can be very slow, because what is happening here is that we are nucleating calcium carbonate before all the mixing has been completed. Therefore, we know that there are local variations in the pH of the aqueous solution, which are taking place while we are mixing both solutions. And we know, it is very well known, that this amorphous phase can have different type of structures at the local scale. And what I mean with this is that it has been demonstrated that this ACC phase can have a more calcite type, a more batrite type structure. Okay, so this structure depends on the pH at which this amorphous phase form. Okay, so that's the reason small variations in pH can completely change the crystallization pathway of calcium carbonates. Second factor, foreign ions and molecules. These ions and molecules can sit on the surface of the amorphous and crystalline phases, and also can enter sometimes into their structures. So when these ions are in solution, what happens is that they can decrease the availability of other ions that amorphous calcium carbonate need to take from solution. For example, they can decrease the availability of calcium, and that can affect the properties of the amorphous calcium carbonate, and therefore that can affect, for example, the kinetics of dissolution and transformation into crystalline carbonates. Therefore, these ions are very important because they affect the kinetics mechanisms and pathways of calcium magnesium carbonate crystallization. And in particular, what I'm going to focus now is in the technique I mentioned before, named PDF, or per distribution function. This is a technique that is very useful because it can show us some data which is key to understand the structure of amorphous materials at the atomic scale. And in particular, this technique is useful because it shows us the probability of finding two ions at some particular positions or distances in the system. Therefore, it's very useful to determine distances between calcium ions, or calcium carbon, or oxygen hydrogen, and so on. Okay, so here you can see one diagram that is showing you two main variables. This horizontal one, this is the distance in angstroms, and this G of R represents the probability of finding some atomic pairs at some specific distances. So here you have higher probability of finding atomic pairs at these or those distances. So first thing you can see here is that in the case of the amorphous phase, amorphous calcium carbonate, which is this black function here, and the degree of order is like a very short. It happens, order is only happening at very short scale, while crystalline carbonates are ordered at longer scales. Okay, so in the case of the amorphous phase, there is no long range order. The order is always shorter than 15 angstrom. And what we do with this technique is to study this ordering of the amorphous phase when it is interacting with different organics or inorganics. We study the solids. And for example, I'm going to show you some details here. This is again the same thing. We are focusing only on the amorphous phase, and what you can see here is the probability of finding atoms at some particular distances. For example, this distance here corresponds to the calcium-oxygen pair. This distance here corresponds to the calcium-calcium pair, and also this one. Okay, and here what you see is 3 different amorphous calcium carbonates produced in the presence of aspartic acid, glycine, and one which is pure. And you can see that the structure at the atomic scale is essentially the same. So they have not changed in the structure. But, for example, when we're playing with some particular ions, for example, magnesium or strontium, this structure can change. So you can see here that these distances of the calcium-oxygen atoms are increasing when you have more strontium, more magnesium. And this is what you see also here. So what we see is that foreign ions can change the interatomic distances in the structure of this amorphous calcium carbonate. So they change the structure of this solid at the local or the atomic scale. And this is translated into different thermodynamic properties. Also, one thing that happens is that supersaturation can increase the incorporation of these ions. And I'm showing you here one example. Here you have magnesium in solution relative to calcium. For example, here you will have 50% calcium, 50% magnesium. And here you have magnesium in percentage incorporated into the solid. Okay, in amorphous calcium carbonate. So, for example, when you have 50% calcium, 50% magnesium in solution at 100 millimolar concentration, you incorporate something like 25%. At 500 millimolar, you incorporate nearly 40%, and at 1000 millimolar, you incorporate nearly 50%. So, supersaturation increases the amount of the proportion of specific ions inside the structure of the amorphous phase. And this is translated into changes, for example, in solubility and other thermodynamic properties. So depending on the amount of magnesium you have in the solid, the solubility of the magnesium rich amorphous calcium carbonate can change. So this is the logarithm of the solubility product, and what you can see here is how the solubility is changing depending on the amount of magnesium in the solid. Third parameter is the ionic potential of cations versus temperature. This is very important because these parameters affect the dehydration kinetics of the amorphous phase prior to its breakdown, and therefore the kinetics of ACC dissolution. So the dissolution of amorphous calcium carbonate is directly linked to the polymorph selection of the crystalline calcium carbonates and to the mechanisms of crystallization. And I'm going to show you some examples with calcium magnesium carbonates. So what happens when you have pure calcium carbonate? What happens is that you can have an amorphous phase like amorphous calcium carbonate, which breaks down quickly in less than 2 minutes, and then baterite forms. This is quick growth of baterite by asteroidic growth that happens in a few minutes. When you start adding magnesium to the aqueous solution and then you mix your solutions, what happens is that this amorphous phase is more stable. Baterite will not form, and what will happen is that magnesium calcite will crystallize after a longer induction time. Then, if you play with higher magnesium calcium ratios in your solution, what will happen after mixing is that you will not form magnesium calcite. You will form monohydrocalcite, and this is a process that can take place in half a day and involves different stages of crystallization. And the amorphous phase here can be stable for nearly 1 hour. At higher contents of magnesium, you also will form monohydrocalcite, but only by applying temperature you can produce dolomite. This requires higher temperatures, higher than 60 degrees C, and here you see one example in which you can see the crystallization of dolomite after the breakdown of the amorphous precursor in a few minutes at 180 degrees C. Why is this happening? Well, this depends on one very important variable, which is the ionic potential. The ionic potential can be calculated by dividing the charge of the cation, in this case it is +2 for both cations, by the ionic radius. And in the case of calcium magnesium, magnesium has a higher ionic potential because it has a smaller ionic radius. Okay, so one thing which is very important is to link this ionic potential to the hydration cells. All these ions in solution have hydration cells. They have water molecules around. And the energy required to dehydrate these ions is directly proportional to the ionic potential of the cation. Okay, so the higher the ionic potential, the higher the energy, or therefore the higher the temperature, which is needed to dehydrate this amorphous phase, this cation. And this is exactly linked to amorphous phases. Amorphous phases are hydrated compounds. For example, amorphous calcium carbonate has 1 mole of water per mole of calcium carbonate. And therefore, what happens is that as the cations are surrounded by water molecules, the kinetics of dehydration of the amorphous phase will depend on the ionic potentials of the cations in the solid. So they are not going to behave the same way if they contain calcium only or calcium and magnesium. And because of that, the kinetics of crystal growth, the mechanisms of growth and the morphology of the solids will depend on how fast this amorphous phase breaks down, or in other words, on how much energy we can provide. And I can show you another example here. These crystals, they are both monohydrocalcite. The first one formed at 21 degrees C. The second one formed at 40 degrees C. In the first one, we have seen a slow, classical growth in which the crystals are growing very slowly and they are nanometer size. Okay? The second one at 40 degrees C at higher temperature formed by spherotic growth. This crystallization by spherotic growth requires a faster breakdown of the amorphous phase, because if the amorphous phase breaks down very quickly, supersaturation in solution increases very rapidly, and therefore what happens is that spherotic growth can be promoted. This will not happen when you have low temperature, because it's much more difficult to dehydrate the cation in the amorphous precursor. And this is what we see for many phases. What I mean is that if we provide enough temperature, enough energy, it's possible to break down the amorphous phase very quickly and to crystallize calcium magnesium carbonates via spherotic growth. And this is what we can see here. You can see here calcite, batrite, aragonite, magnesium calcite, monohydrocalcite, dolomite. All of them form via spherotic growth. And this is happening because we have provided enough temperature to break down the amorphous phase very quickly and to promote spherotic growth by increasing rapidly supersaturation levels in solution. Okay. Does this happen in nature? Yes, it happens, and in fact we have seen spherotic growth, for example, happening in dolomite, in aragonite, in batrite, in monohydrocalcite, and this is telling us that many of the processes that we are mimicking in the lab can happen in nature. So learning how calcium carbonate form using laboratory, conventional or synchrotron-based techniques can be very useful to understand what is happening in nature during biomineralisation processes, or also during abiotic formation of calcium carbonate. Okay, I'm now moving to the second part of this talk, which is more focused on synthetic calcium carbonate, and in particular with a focus on industry, because I want to explain you how we can obtain nanocalcite. Nanocalcite can be very interesting because if we have nanoparticles, they will have a very high surface area, and we can envision two ways of producing nanocalcite. The first one will be by size reduction. We can take a big calcite crystal and start breaking it in pieces to obtain nanocalcite. The problem is that in theory that should be very simple, but we can produce microparticles, but we don't reach the nano size. Second thing is that what happens is that we usually contaminate the material with the tools that we use to break that. Okay, so therefore this method is not really useful. Another method will be the opposite, to crystallize calcite from solution. And yes, we can go in this direction, but usually the problem that happens is that when we're crystallizing calcite, we produce crystals which are bigger than nano. We produce microcrystals. And if we want to produce nanocrystals, what we have to do is to use additives, organics or inorganics, that will prevent growth of this mineral. Okay? Because otherwise it's very difficult to retain the size. Alright, so the problem here is that we want to obtain nanocalcite that is clean. We don't want to have additives, or we don't want to have contaminants. We only want to have calcite. So essentially, problem, there is no good and let's say fast and cheap method to produce nanocalcite, and what we want is high surface area, or in other words, very tiny calcite grains. Also, pure calcium carbonate, no other ions there. Also, pure calcite, no other polymorphs like aragonite or batrite. Also, a clean surface, we don't want to have inorganics or organics attached to the surface. Therefore, the surface of calcite will have the properties we want. It can be used to trap, for example, pollutant. It may be possible that this calcite exists in nature, and in fact it happens. Nanocalcite exists in nature. For example, coccoliths found in places like this are nanocrystals. But the problem is that despite they have a very high surface area, this is not pure calcite. It contains other ions like strontium, magnesium, and organics on the surface. So therefore, the surface properties of this nanocalcite is completely different compared to pure calcite. It may be that we can use commercial calcite, but commercial calcite is usually bigger. It has a smaller surface area and is usually not pure. What I mean is that it has some organics which are used to prevent degradation of the particles and to prevent the formation of agglomerates. Okay, and perhaps what we can do is to produce this in the lab. There are methods to produce nanocalcite. The typical one is by bubbling CO2 in a solution with calcium hydroxide, and yes, sometimes you can produce nanocalcite. There is a problem of reproducibility, but very often the problem with this method is that you produce nanocalcite, but inside the grains you still have some remains of calcium hydroxide. So essentially, the usual two methods are problematic. The method of bubbling CO2 is simple, quick, but there are inclusions of calcium hydroxide in the nanocalcite, and there are usually reproducibility problems. So this nanocalcite is not really good because it has inclusions. Okay. Also, the use of inhibitors or dispersion agents is not ideal because these are organics that will sit on the surface of calcite and prevent further growth. But if you want to use this calcite for example, for remediation, for the removal of contaminants from solution, this calcite will not have the same surface properties compared to pure calcite. So here, what we want is to produce calcite with very small sizes and very clean surfaces. So let's see the problems that we have. Okay, first thing is that we don't want to have foreign ions. We only want to have calcium and carbonate and not ions like strontium or magnesium that will change the properties of our calcite. Also, we don't want to have ions on mineral surfaces. I mean, we don't want to have inorganics or organics sorb on the surface of calcite. Also, we don't want to have inclusions, dissolved or non-dissolved reagents, because that will change the properties, the dissolution, the equilibrium pH at which calcite is with water. Okay. Also, we don't want to have other polymorphs. We don't want to have aragonite or batrite or anything different than calcite. And we don't want to have aggregation or spherulitic growth because we need nanoparticles to get a high surface area, and therefore we need to have isolated particles that they don't aggregate. Okay? So essentially, all these problems can affect the surface area of the product and the properties. Okay? So I'm going to show you the method that we use to produce nanocalcite, and this is a lesson for you to understand how important it is to control crystallization of calcium carbonates and how important it is to understand the mechanism of crystallization of these materials. Okay, this method is quick and it is made of two different parts. Part A is nucleation from solutions. So what we do is to mix two solutions of calcium chloride, sodium carbonate, and nucleation of amorphous calcium carbonate happens instantaneously. So therefore what we do is we filter these solutions using vacuum filtration, and then we add some isopropanol. This isopropanol will prevent interstitial water to be there. Interstitial water is dragged, and what happens there is that we only have particles and the isopropanol will evaporate very quickly. Okay. Then what we do is we put these nanoparticles inside a crucible that goes inside a furnace at 450 degrees C. And this amorphous calcium carbonate transforms via dehydration into nanocalcite. Okay? The first part is very quick, needs to be done in less than 2 minutes. The second part only requires half an hour. So, just to summarize. Okay, we mix solutions, we get a supersaturated solution with calcium carbonate. This solution will contain amorphous calcium carbonate, and then we do filtration, we quench the reaction by using isopropanol, and we dry at 450 degrees C because that will evaporate the isopropanol, also it will transform the amorphous calcium carbonate into calcite. So there will be no remains of isopropanol, and all the amorphous phase will be 100% calcium carbonate calcite. Okay, so let's see the results. First thing, let's try to see the surface area. Okay, this graph shows you the concentration of the starting solutions, and here you can see the surface area in square meters per gram. And what we see is that if we use solutions from 5 to 10 millimolar, we get an increase in surface area. Concentrations above 10 millimolar result in a lower surface area. So I will tell you why this is happening in a moment. All right, if we take this nanocalcite with the highest surface area, what will happen is that we can put this nanocalcite in water and the surface area will decrease. In 9 days, it decreases in half. In 16 days, it decreases like to 20% or 15%, okay, to 4 square meters per gram. So let's see how this nanocalcite looks like. Okay, the particles, when they are taken from the oven, they look like that. So here you have nanocalcite with 14, 16, 18, 23, and maximum 24 square meters per gram. Okay, so these are particles which are spherical or pseudospherical, and this is the highest surface area of calcite we can get. So when we get these particles, we can put them in water, and when we put them in water, in pure water, what will happen is that they will start becoming rhombohedral. You will get rhombohedral calcite particles, which are like the typical calcite that we'll see in other experiments or in the field. Okay, it has the usual calcite shape. And with time, in 9 days, the particles grow and therefore the surface area decreases. Okay, so this is a method that works very nicely because we can produce nanocalcite which is spherical or rhombohedral, and we can select the surface area by controlling the concentration and then, if we need to, the amount of time in water. Okay, this is a comparison of this calcite with nanocalcite obtained in our experiment. So this picture here is showing calcite that you can buy commercially. This is a typical commercial calcite that you can buy from different suppliers. Okay, this has a surface area of 0.2 square meters per gram. This is a typical calcite that forms after baterites in the laboratory, which is very easy to make. You mix solutions and you wait, and after a few hours you will have these calcite crystals, 2 square meters per gram. Here you have the usual shapes of coccoliths. This is nanocalcite that has 2 to 8 square meters per gram, but the problem with this calcite, as I told you before, is that this contains some strontium, magnesium, and some organics. So the behavior of the surface properties are completely different. And here what you see is our 3 nanocalcites. So this is the smaller surface area of spherical nanocalcite. This is the larger surface area of our nanocalcite, and this is what you get after the interaction of this nanocalcite with water. Okay, so you can compare the sizes here because the scale bar is the same for all of them. So this shows you how important it is to control the mechanisms of nucleation and growth, and to understand what is happening in our system to be able to obtain calcite with the properties that we want, with the surface area in this case that we want. This is showing us that there is only calcium carbonate. You can see here the pattern of amorphous calcium carbonate, then the pattern of crystalline calcite. This peak width changes as a function of surface area, that means of particle size, and this is something expected when you have nanoparticles, that the width of the peak increases, and it looks that in our system we only have 100% calcite. Is a surface clean? Well, first thing, that we were using isopropanol. What happens when we put isopropanol in the oven? What happens is that it transforms into water and CO2. So we need to check if our surface of calcite is clean, and for that purpose what we did is to use X-ray photoelectron spectroscopy. And you can see here the patterns of 2 different calcites, of 2 different nanocalcites, and they consist of only calcite. The only difference here are these 2 peaks here. This peak on the top, the brightest one, corresponds to typical carbon in carbonate bonds, and this second one is adventitious carbon that happened in one sample, and this is something that usually happens in many conventional calcites. It's not a problem, and that demonstrates that our surface is extremely clean. Okay, we don't have any remains of the isopropanol. So, in summary, okay, our nanocalcite works because it has a very high surface area, and we can target the surface area we want. It is pure calcium carbonate, it is only calcite, and it has a clean surface. So it is fantastic, as a product to do experiments later on. Okay, and here what we do is to control what happens during nucleation and growth. So we use this kind of combination method. We nucleate from solution, and then we do solid state crystallization. So we can control later on the shape and size when we put calcite in contact with water and we grow it via oswald ripening. Okay, so we can avoid slow crystal growth, aggregation, spherulitic growth by using this method, because all these methods will produce bigger calcite crystals with smaller surface areas. So essentially, what you have seen here is that you have calcium carbonate solution, amorphous phases, crystalline carbonates, but what we do is to follow a different line that we control. We produce amorphous calcium carbonate, then by a solid state, we transform this calcium carbonate into nanocalcite, and if we want to, we can grow this nanocalcite via oswald ripening to make bigger calcite crystals as big as we want. So finally, there are three main variables that affect calcium carbonate crystallization. First one is the pH. Second one are the foreign ions present in the aqueous solution, even as when they are trace elements, and also very important, the ionic potential of the cations versus the temperature. So if we control these variables, we can target the crystallization of calcium carbonate with specific properties. So this is very interesting for us to understand what happens in nature and how we can control calcium carbonate crystallization, how we can produce industrial calcium carbonate for many different purposes. Obviously, calcium carbonate is a very complex system, or in general carbonates. So ideally, we will need a unified crystallization model, and this is something that will take plenty of time to be developed. That will involve research by many people in the future years. Thank you very much, and thank you to the European Association of Geochemistry for inviting me to give this talk.